Thank you so much. It's such a pleasure to be here. Thank you for the invitation. It was lovely to have a reunion yesterday night. It's as my former life, I was doing human genetics. I'm not an active member of the V2F community, but it's such a pleasure to get to meet all of you today. I was honored to come and share with you some of the work, resources, and tools we are developing as part of our work done in the Human Atlas Initiative, where I'm currently the vice co-chair. And so while I do, my favorite cells are immune cells, I'm hoping that some of the frameworks I will present can be useful to your community here. These are my financial disclosures. And so this is a quick outline, so you know where I'm going to go with the presentation. I want to tell you about some of our first large human blood cell atlas, which is compiling over 40 million cells across 40 different conditions, some of the challenges we had to overcome to integrate so many different data sets, how we're tackling sanitation challenges, and how we're deriving novel biological insights. And I promise I have some snippets that are relevant to variant to function in there. So kind of bear with me. So why did we start with blood, right? You can tell it's the most boring tissue. Actually, when you think about translating our findings clinically, it is a window in human health. It is easily sampled, it's biobank everywhere in the world across all clinical trials, very easy to collect in the clinic. When you think about methods development for single-cell multiomics, in this case we're going to talk a lot about single-cell RNA sequencing, these are the largest data sets that are out there right now. And so there's an opportunity to try to go beyond every individual studies that have been published, bringing them together to derive more powerful insights. And so what we hypothesized coming through this project is that if we could integrate as many data sets as possible, we would saturate the signal that can be detected in the blood with the hypothesis that there's going to be a finite number of distinct cell subset identity in the blood, and some of them will be conserved across disease, and that could help you hone in on some of these conserved cellular programs. And so I'm going to show you some highlights of how we've been working towards 40 million cells. I'm like the hungry hippo. I always want more cells. And so I told my lab it would be so much better if I think some of them are in the other room. There's witnesses here. I'd like 15 million. We're working towards it across over 5,000 samples and 40 different immune conditions. And I'll walk you through, in case there's some immune lovers in the room, of the different data sets we've compiled. So starting with some 4 large integrated immune atlases. One of the ones we're currently wrapping up, which was tied to a lot of computational methods development, is tied to the Human Cell Atlas. In case you've never heard of it, it's an international endeavor that includes over 104 countries across 4,000 members across 104 countries. And our mission is to create a comprehensive reference map of all the cell types and properties of human cells and human body and health and eventually disease. And so these are the members of our version 1 Immune Cell Atlas. We have groups represented across Singapore, UK, Germany, Spain, and of course Boston, with trainees across all of these different sites. And some of our initial goals are to establish a detailed reference of all circulating cells, so that when we move to tissue, which we are moving to tissue, it becomes very easy to distinguish resident from circulating cells. It turns out to not be trivial to integrate so many data sets, so we're optimizing computational frameworks to do so. And I don't know how it is for other communities, but immunologists are very passionate about how we call cell types. And so there's actually a need to come together with the same data set to have this conversation in a more productive way. I'm going to show you how we're doing this. And of course, we want to support downstream biological queries. Some of our goal of Human Cell Atlas is to have a comprehensive, globally represented atlas, so we actually have data sets from all around the world in there, so you can also see how genetic diversity can shape cellular identity. So I should say the first one, let me go back if I'm capable of doing that, focuses on healthy and COVID. That's mostly because we needed data sets with the same pathology across multiple institutions. We've also teamed up with Dr. Holger Heinz from SINAC in Barcelona, who has now this data set in preprint, and their whole goal is to build a disease classifier using blood. So if you select a few drops of blood, can we tell which disease you have? So we've integrated their data. There's also beautiful studies that have been published in preprint from the Allen Institute and from UNL, where we've also integrated their data. And that's how we get to 29 million cells across over 3,000 donors. We've also went through everything that has been published in the literature and gathered data sets across healthy, infectious disease, autoimmune disease, and adverse events to checkpoint. We got another 5 million cells. And my lab contributed another 8 million cells of unpublished data across healthy infectious disease, autoimmune disease, and adverse events to checkpoint. And that's the breakdown on the, your left, of the different groups of disease, autoimmune disease, infectious, adverse events. You'll notice we don't have heme malignancy. We're really focused on trying to understand the biology tied to the host. And so with that, we have data across single-cell RNA sequencing, we have surface protein, we have single-cell TCR sequencing, and I'm sure somebody's going to ask me, where's the genotypes? This study was not designed, it's retrospective data, so we don't have a lot of genotype data, but I'm going to show you how we can still try to pinpoint cellular contacts that may be relevant to GWAS. And this is the amazing team we have here, bridging between the Broad Institute and Mass General Hospital. It's an effort that I'm co-leading here with Gary Reynolds in leading the integration and leading the data generation across all of these diseases. And so we have 4 main objectives. The first one is to create that foundational resources, right? We want to be able to create a resource that anybody can use to map annotation for circulating cells moving forward, so you don't have to re-annotate over and over again. We also want to benchmark better ways of doing cell annotation. From a translational perspective, we want to try to start mapping the cellular molecular programs that are shared across disease. I realize this is blood, but you might be surprised. I'm going to show you that even in blood, where the disease is not unfolding, some of them are systemic disease, you do find these shared programs, and start thinking about potential targets here. So I'm going to spend the next few slides telling you how we optimize our computational framework. It's actually not trivial to do, so it would be so much easier if we had generated these 40 million cells in a single institute. But this is hundreds of studies. So we had to optimize the framework using a subset of studies, and we picked autoimmune disease to do so. Why so? Well, they're tractable cell number. We know the biology, so we know some ground truth. There was moderate batch effect because we generated a lot of data locally. And we had to figure this out because the current computational workflow that existed when we started this project couldn't handle that many cells. And so why, for those of you who don't think like me about autoimmune disease all the time, why start there? Well, we already know there's a lot of commonalities across autoimmune disease. We know in terms of organ involvement, there's a lot of organs that are involved in different autoimmune disease. We actually know that some diseases respond to the same treatment, also hinting that there's some commonalities. And for all of you who with the V2F community, you probably already know that there's a lot of genetic variants that are shared across the disease, also pointing out to commonalities. And so this is where we started. What were the main challenges? So first, integration. We spent over 2 years on this. I promise this is not going to bore you with this because it's very dry. But we're going to release all of our code so that people can see how we've done this. We spent a lot of time optimizing how we do quality controls for raw data and processing and lineage-specific processing. How do you optimize integration strategy in terms of feature selection when you have to deal with disease and normal tissue? So thinking about highly variable gene selection. We actually didn't take the first integration tool out of the box. We actually benchmarked 5 different integration strategies, optimized different parameters, only on to 2 of them, Harmony and SCVI, and the winner for us has been Harmony. And then we did hyperparameter tuning on all of these features here. And to take a decision, we had to develop both qualitative and quantitative assessments. So on the left, you have quantitative assessments. So that meant like looking at distribution of source data set, chemistry, cell range version, gender, donor, and so forth. We also wanted to make sure that the biology was conserved. So we know so much about immune cells, it's a great place to start. So for example, we could tell that some of these plasma cells, we couldn't split the IgG from the IgA plasma cells. So for example, this would be an example of, an embedding would be rejecting. We use the SCIB framework developed by the TICE lab, but we also have been developing new metrics that really assess biological conservation. For example, if you know groups of cells like Tregs are supposed to co-localize, you can actually quantify this and kind of speed up identifying the right set of parameters. So this is the extent I'm going to bore you with systemic assessment of integration. But if you're curious and trying to tackle this, please do reach out. And once we have some embedding, so this is an example of monocytes and a type of dendritic cells where we have unique markers, we just make sure that across all the different cell subsets represented here. On the x-axis, we have good distribution across disease, type of, in this case, 10x chemistries, because we had over 6 different types, and across all studies. So this is an example of like a final product here. The second challenge we had was scalability in every sense of the word, but in this case, I'm going to talk about annotation. If you start processing tens of millions of cells, you cannot easily annotate this manually. The traditional pipelines are resource limited. And so we needed to kind of find a way in between what immunologists would accept and what computational biologists would be willing to work with. And so we developed SC-Tiger. You can imagine the lab spent a lot of time thinking about the acronym here. It's for single-cell transcriptional immunoprofiling guided by expert reasoning. And so the way it works is, there's a lot that is known already about the immune cells. Nobody's going to come and argue with me about what is a naive T cells or an effector T cells. But we want to be able to quickly get to the non-contentious label and then do the discovery of cells. So we built a classifier, and this is not exactly the classifier because it didn't fit on the slide. But essentially, you can split based on lineage using surface markers, and we've developed sets of gene-programmed genes so that within minutes, we can get to non-contentious buckets of cells, like the naive, the effector memory, and so forth. And from there, we do unbiased clustering. That seems very logical for those of us that do immunology, but these tools were not in place. And so we did our computational biologists against SC-Tiger. They got to the same conclusion, except that computational biologists took 2 weeks shuffling cells between bucket, and SC-Tiger took a day. And so this is what we've implemented. So SC-Tiger in action, if you put this on half of our cells, 20 million cells across 4,400 samples and a little bit more than 40 conditions, this is what it looks like. So you can start with the lineage level, the top level of the hierarchy. We can find our lineage. You can do another level of annotation and a third level of annotation, at which point, these are the different number of cells per cell subset. You can also look at them across the different conditions. And once we get to this point, this is where we're ready to do cell type discovery. And so I'm going to do a little plug here for another tool we've been developing. For sake of time, I don't have to do demos, but I want to tell you about how we're going to resolve annotation. So, if you give me 5 million T cells to annotate and I give them to a friend of mine, the same 5 million T cells, I promise you we're going to get to different results and different annotation and different names. We might regroup the cells the same way, we might not. We might give them the same markers, we may not. It's a huge problem in the community. At least I can talk about the immune cell atlas community. So same data, same pipeline, different answers. How do we resolve this? And so we're launching a cell annotation jamboree. If you're interested, you're welcome to join. I'm going to show you about the platform we developed for this. Everybody's going to be getting the same 10 to 15 million cells, and you will have to provide an annotation solution. We also have a challenge tied to providing solution for cell spectrum characterization, because these cells are not binary in nature. And there's always a question of how many cells do you really need to profile to identify all new cell types. Now we have so many cells, we can answer this question of how many donors, technologies, processing modalities you need to identify all predicted cell types. Over the last 4 years, with support from Schmidt Future at the Broad Institute, we've actually developed a cell annotation platform. This is a user-friendly interface that is meant to create, explore, and store annotation. We have created the infrastructure to accumulate and analyze annotation, as I'm going to show you. We're developing products to offer automated annotation, and it's meant to get the community to work towards consensus annotation. So how does it work? First, you need to move annotation to be subjective to a more objective definition. So we've defined clearly 12 annotation metadata fields that are listed here. So you go beyond the cell label, the full name. We want the canonical markers, but we also want the markers that you use to define a cell type. We want to know if there's an ontology term that exists or what is the closest term. And these are extremely well-defined metadata field. And this provides you the necessary context to more consistently and reproducibly construct atlases. But also, by having everybody provide these annotation metadata fields, we can now properly compare and contrast annotation between groups and provide feedback. For sake of time, I won't do a demo, but it's actually an interactive tool. You can do rounds of votes, you can agree, you can disagree, you can split, merge clusters, provide your annotation. You don't need to know how to code. And these are the type of modules you can do. It's a bit like a Google Doc. If you wish, you can work in private space and invite anybody you wish to work with you. This is not just pre-computed. You can actually do live compute on this browser, which makes this portal different than other portals. And so you could come, and with our widget, you could actually select cells. It will recompute gene expression. It will recompute marker genes for you. We can do gene signatures, differential expression. We're going to be implementing automated annotation. You can look at a relationship between data sets. And ultimately, the goal is to have the equivalent of gene card and summarize annotation post feedback. So this is a quick plugin. If you want to hear more and see demos, do reach out. Okay, I'm going to now move to the more interesting part, which I'm sure is more interesting to you, but I wanted to kind of plug in what we're up to. So let's move to biology. And so, okay, we have all these millions of cells. What can you learn from this? Well, of course, we could try to understand if there's shift in abundance across disease, gene programs, or so much more. And so what did we find? These are examples of analysis we can do. So abundance, risk variance cellular context, which I'm sure is the only vignette you want to hear about. This year, repertoire analysis and gene programs, but let me tell you a bit about each. For abundance analysis, again, this is where our paths for autoimmune disease. Broadly, can we find some cell subset that shift in similar way across disease? And I will admit that this is a bit generalized, and one should ask me how these samples were selected later in the question period, so I can expand on this. But PDCs, we can see in several of our severe patients are up in many of these autoimmune disease. Tregs are done in many of these autoimmune diseases, and some of it is known, so it's a good way of assessing how good our pipelines are. But you can also find subsets we didn't know about that are associated with specific disease or multiple disease. So for example, we could find proliferating plasma cell expanded specifically in lupus here in blue. I should say the legend here is here on the far right. We also find a new B-cell subset we didn't know about that was specifically expanded in lupus, rheumatoid arthritis, and type 1 diabetes. It has very specific markers, and when we look at the embedding, this is this specific cluster, which we know very little about, that was specifically associated with expanding this disease, which we're now following up on. I know this group doesn't need any introduction to this. And so in the context of autoimmune disease, there's been hundreds of loci associated with autoimmune disease. You can ask very broadly, what have we learned? We've learned a great deal that there's a lot of these risk variants have very low risk, relative risk associated with them, but they tend to be enriched in a similar biological pathway. And so the question here is, what is the relevant cellular context to study? And as opposed to targeting some of these specific gene variants that have very small relative risk, could we think about the cellular phenotype that we could be targeting instead? So how can we look at this in the context of the data sets we are gathering? So first, obviously you collate your risk variants with the highest predicted effect. In this case, for sake of proof of principle, we use those from OpenTarget. And we use a method developed by Zhang et al., published in Nature Genetics in 2022, to identify the cell types in which these variants is predicted to be active using what they developed being the single-cell disease relevance score. So the way it works is that you assess whether a given cell has excess expression level of a set of putative risk variants from any GWAS you wish, and we use a matched empirical null distribution to estimate the well-calibrated p-value, which you can compute based on the pool normalized control score across all gene sets. So for sake of demo here, I can show you that we see enrichment of some of these variants associated with lupus in PDCs. Now, a lot of these genes tied to different disease susceptibility are not independent from each other, right? They're co-expressed with other genes. So here I'm showing you some example with rheumatoid arthritis, where we look at the co-expression of the different risk genes and identified several modules across our data sets. And then we wanted to know where these modules were expressed, if they were cell-specific. And we saw different patterns. For example, if I take Program 5, which is here, you can see it's expressed across most cells, and I'm going to show you more detail next slides. If you look at program 3 here, it's actually a little bit more specific to myeloid cells, and program 2 here is specific to a very particular cell types, and I'm just going to zoom in to show you. So I mentioned program 5 is across all cell types. Here, this is not a fine-grained annotation. This is just made out of the path of optimizing a framework. You have the different cell type organized by lineage, and you have a dot plot. The darker the plot, the bigger it is, the more it is expressed in a particular program, so you can see it's expressed everywhere. Program 3, as I showed you, was mostly myeloid, and then program 2 is very specific to one subset we've defined that had very inflammatory gene program. And if you zoom in on program 2, which is here, it's this particular subset, and it had a lot of specific markers, including TNF, IL-6, IL-1 beta, specifically expressed here. Just to kind of show you the power of some of these analyses. I may not have the genotype of these patients, but I can still tell you what could be relevant here. Now, I'm also interested to know if there's enrichment that are shared across multiple diseases. And I can do very similar analysis here. And in this case, Tregs comes up as being enriched for these susceptibility variants across all disease. I can, here I'm showing some of these different diseases, some of these key genes that are up across, let's say, lupus, type 1 diabetes, and RA. And I can find some of these genes driving these analyses, including some of these key ISGs, interferon-stimulating genes, and I can look at where they land in the context of our embedding. Not only do they land in the Tregs, they actually land into a very particular subset of Tregs that we didn't know about. And so it helps us hone in onto what cell subset we should be doing some follow-up studies on. So I'm showing you some of the key genes here. I should have mentioned none of this is published, but we're sharing this so that we're hoping to motivate the community to embrace some of these tools. And finally, I'll skip TCR for sake of time, but gene program very quickly. So many different ways of looking at gene programs. I'm going to show you one vignette using consensus non-negative matrix factorization, or CNMF here, where we're trying to define gene modules that are tied to both cellular identity and factor activities. I'm using the myeloid cell as my example here. And so, as we try to map the activities across the different cell type, it's a move, we're simplifying it, but a combination between these programs that are going to be tied to the true cell identity, and those that are tied to its activity. And so in terms of this true identity, I'm showing you these different programs. These are marker genes that define these cell types, not shocking, but we're interested in the gene programs that are tied to function, that are across many, many subsets. And so, for example, we find these IgG program in IgG4 disease, lupus pediatric, which is known. But you know how I told you about these IL-1 beta monocytes? When they actually come up as a gene program. And you can actually find them specifically in IgG4, Lyme arthritis, and other disease. So that's another way of kind of coming together. So to summarize, I showed you some generalized strategies to define shared and distinct programs. We finalized integration of these 20 million cells, and we're actually using the other 20 million cells we have to see how generalizable our findings are. We've developed the cell annotation platform. I've mostly went over it, but if you want to hear more about it, do reach out to me. I've shown you that with our framework, we can find shift and abundance that are shared and distinct across disease. We can find certain context for susceptibility genes. I didn't cover the TCR, but we can also find TCR that share across disease along with shared programs. And so you can imagine what we're going to be doing next. If you're interested about the annotation jamboree, do reach out. And we're going to be releasing all our code on GitHub, so if some of it is useful to you, I'll be delighted to team up. And finally, I tried to show pictures of the amazing team that has been involved. This is my partner in crime, Gary Reynolds, for doing this, the cell annotation platform team, the whole lab, actually. More than half the lab is working on this, the funders, and more importantly, the patients and their family, none of which would be possible. It wouldn't be possible to do this work without their partnerships. Thank you for your attention. This is working.
Okay, good. I'm interested in how you you started out by saying your assumption is there's a finite number of cell programs. So I wonder, with the current atlas you have, with about 30 million cells, how close are you to being able to estimate sort of the number of potential finite number, and how does it change in terms of when you compare it across answers? If you look across ancestries and perturbations and stuff.
That is a good question. So I should say cell subset. I don't know about the cell programs until we model it. And so we wanted to know after how many diseases do we stop seeing new cell subset emerge, because if you start grouping other types of disease, at some point there's only so many cell subsets you can identify, and we can downsample our data sets to see when we start saturating. And only in the context of course, what I presented, which is infectious, autoimmune disease, normal, pediatric and adult. I didn't mention this. We have pediatric and adult that are all non-treated. Then we don't see more signal. Of course, once you add pre-post treatment, it's a different story. You're completely correct in terms of ancestry. As we add more ancestry, we may see a bit more diversity. I didn't cover this, but we actually have representative samples from almost everywhere in the world, but we definitely are skewed towards Caucasian. So the next iteration will actually add another 3,000 samples from South America and 2,000 samples from Africa that will help with this. And then we'll be able to estimate.
Great, thank you. Ben?
Lovely talk, Chloe. My question's on the cell annotation, and particularly how you're dealing with the sort of subset of transcripts that you're actually sampling in the single cell technologies, because as I understand it, we're getting maybe 15 to 25% of the transcripts per cell. And I noticed in your sort of flow charts, there were required genes for cell identity, but given the amount of missing data, do you worry about annotation problems cropping up there, or is the kind of overwhelming bulk of the data sufficient to still get you to the right place?
I love that question, Neil. So first I should mention that for the cell annotation, we also added over 200 surface protein, because immunologists love surface protein, and that's how we can relate the gene signature to surface protein and relate the legacy knowledge. We've also included TCR and BCR data because it can help identify specific T cell subset. Now, of course, if you look at one cell at a time in human data, you're right that we're capturing 1,500 to 2,000 genes. Some of it is dropout, but some of it is also you're looking at primary cells. So at any point in time, you don't express all genes. And so once we've defined groups of cells with similar features that you think have unique identity, you actually can do pseudobulk, and then you capture a lot more genes. And so that's a way around it. And then the single cell community is already doing a lot of annotation. It's all over the place. And so there's a need to start agreeing, maybe you and I would call the same cells two different ways, but it'd be great to know that we're calling the same cells two different ways. We can build that catalog of synonyms, and that's kind of what this platform will also help us get to. Yeah.
Great. We'll have one final question. Liz.
Thank you. Chloe, this is amazing. What you've built is such a service for the community. I wanted to ask you about multi-omics. So some of these data sets also have ATAC. How's the HCA thinking about incorporating ATAC? Because it would be incredibly useful to build a similar resource and leverage your cell type annotation platforms for ATAC-seq too.
Yeah, so the CAP can be adapted to any species and any modality. And then in terms of HC and multi-omics, we're all for it. The reason you saw so many single cells is that we had to start with the modality that was most out there and most scalable and cost-effective, and a lot of it was single cell with protein, which I didn't disclose, and TCR. There's not that many multimodal data set out there, and I think it's also tied to the cost of generating such data at scale. We are actually integrating such data set, but to be completely honest, it's a very small percentage of the data. And as part of HT 2.0 that's now being launched, one of the pillars is to push for multi-omics and trying to find partnerships with industries so we can drop the cost. But it may make more sense to kind of push such effort on very specific contexts or disease or cell subsets where we actually need to map the regulatory landscape. And this is where partnership with this community could kind of help synergize such effort. Thank you for welcoming me. Thank you for your time.
Thanks, Chloe.